Okay, we'll go ahead and get started here. Now, with the content standards for this state, it's pretty heavy invested in genetics, which is what we are doing now. And I would be led to believe if that's what the state thinks we should cover, it's probably going to be on some of your national exams, like your ACT test, okay? So there's a reason why we are going through this, and I don't know if you want to say taking our time, but making sure we have a good understanding of this. Because what do you suppose it means? The higher score you get for an ACT test, the higher number you get for a score on your ACT test, what does that translate to? Anybody know? How many are taking the ACT next year that you are aware at this time? Oh, okay. Do we know what the ACT test is?
Yeah.
Admissions College Test. I think that's what it stands for. I could be wrong, but, and you don't need it for a technical school. So, and I'm just going by what I observed. Nobody either wanted to raise their hand or saying that they are not, and that's fine. Okay? So with this bottom problem, I have a visual aid for you to watch here. But we will put these traits to the terms, whether they're heterozygous or homozygous, because I'm not totally convinced, do we understand the difference between those? Okay, so we're asking for input and we're getting it. Sorry?
Is heterozygous the dominant trait?
No. Okay? So the first thing that you're going to want to do is establish and understand what's dominant, what's recessive, and then make a key. So if you are tuning in later at home, it says what are the phenotypical results of a cross between heterozygous tall and green pea plant, or I should say between a heterozygous tall and green pea plant if it's crossed with a heterozygous tall, heterozygous yellow plant. Tall is dominant over short, and yellow is dominant over green. Okay? So, if we're going to make our key, there should be 4 letters to this. Okay? So, what do you want to establish for the tall trait that is dominant? And you could use this if you wanted to and say that that's tall. You can use any variable that you want. Just like you, with algebra, you usually use x, you can use y, you can use z, you can use any variable, but if you want to choose b or if you want to choose t, that's fine. So that means tall. So then, yes, this means short because it has to be a contrasting trait. Okay? So if you have a tall trait, you have to have short because that's what it's talking about up here. Because here we see tall is dominant over short. So what variable do you want for yellow? Okay? You could use G if you wanted to because green is in there as well. So capital Y is yellow. And then lowercase y is green then. The other contrasting allele on this trait. So when we're talking about these traits, you find them on chromosomes. And these chromosomes are found in the nucleus where all that DNA is located. And I think you would be surprised as well that within the human genome, all of those nucleotides, it's just this difference. It's supposed to be a T, but instead there's an A there. One nucleotide causes that sickle cell anemia. Okay? Just one individual nucleotide. So that's why when we study these genetics, that's important that some people will choose to do genetics counseling. Why would someone want to see a genetics counselor, do you think? It's probably for people who are going to have children. Why would you want to see a genetics counselor, do you think? That's probably the biggest reason that you might do that. If there's any family history, it could be heart disease, okay? It could be diabetes. It could be even the sickle cell trait. It could be a multitude of ones that you can possibly have treated before this goes to fertilization. That's just what I think. I don't want to trade science fiction for this, but this is where we gather all this information. This is where it would have started, even back with Gregor Mendel. Okay, we have our key. Now we see our traits. It says heterozygous tall. What two letters make up that heterozygous tall? There have to be two T's, but what is the combination of T's? Would not be two capitals. One capital, one lower. Okay? So I'll put those over here. Okay? And it's also green. This is not a codominant cross. It's not an incomplete dominant cross. It is a complete dominant cross. Because when you look at next week's schedule, next Tuesday we're going to start talking about codominance. Okay? So that's the example we gave yesterday that you correctly answered. What is codominance?
The cows?
The cattle. That's right. Cattle is a good way of observing a codominant trait. Okay. So what two letters do we use for green then? Because look at our key if we need to. In other words, could we do, would that result in green? It can't. You're right, but why are you right? Because you have this dominant yellow trait in there. Well, let's remove that, and then it must be this combination. That will result in a yellow seed color. Okay? So now it's crossed with another heterozygous tall. So if the one on the left was heterozygous tall, what can we presume about the one on the right? Same thing. And then what can we presume heterozygous yellow? What are the two alleles for that? Capital and lowercase. That's right. Okay. So notice it's just asking for the phenotypical ratio. Unlike this one, it's asking for genotype and phenotype. I don't know if we'll maybe get this one set up. I don't know if we'll get through it or not. Okay, we have our key, we have our cross. Now, let's go ahead. Do you want to work on this on your own or together? Okay. Okay, so, and that's fine. I was asking for input, and we got it. So, give you a few minutes to go ahead and work on this. Okay, so we'll start that now. Are we having problems getting these set up? I mean, if you're not honest, you're not helping yourself out. So we did have a question. How do we know that? Okay, so we try to make sense of this, like for instance, when we talk about factoring. And I don't know if this helps or not, but for me, hopefully looking at it in this manner helps explain this to you, okay? So we had x plus 2 and x plus 3, okay? If we had to distribute that out, okay? You're foiling that out, right? Your first term, you get x what? X squared, right? You take the first times each other. Yeah, x squared, x times x. Okay. Then you do your outside, x plus 3. And then your inside, and then 2x, that's a total of 5x, correct? 5x. Then you do your last term, you're adding what? Adding 6. Okay, and again, we're hoping that when we see this, because you've done this before, we're taking information from here and applying it to this. Okay. So now, just like when you FOIL that, your first term, okay, just like x times x gave you x squared, you do capital T and lowercase y, because that was your first terms in the trait. Okay, then you do your outside one, where you took x times 3, was it? Yes. So here is your outside. Okay, so capital T, lowercase y. Then you do your inside ones here. Lowercase t, lowercase y. That's where this one comes from. And then finally, your last one. Okay, lowercase t and lowercase y, and there it is. The next one, there's a little more variance in there because we don't have a homozygous recessive or trait over here. So we're going to get different types of combinations. Okay? So, what is your first terms here? Maybe rather than having these up here, maybe erase these so we have a little better practice. What is your first term like we did on this side? Capital T and capital Y. Okay? So capital T, capital Y. So your first term here. What is your outside term? Capital T is on the outside, and lowercase y is on the outside. Okay? So here is your outside, these two. Now we go to your inside term, which is lowercase t and no. Capital Y. And then what is your last one? No. Lowercase t and lowercase y. Okay? How many would be led to believe this is where we get stuck, trying to get these across here? Okay? And this is still brand new to you. I was asking for input from the freshmen, and I got the input that you've done these before.
Yeah.
Okay, so you say yes.
But I wasn't here when we did that, so I don't know.
Okay.
We've done it before, but not because new letters. Yeah.
Okay, not this big then.
Yeah. It was like 4 squares.
Oh, so a monohybrid cross, not a dihybrid like this. Okay, but you did it in Del Rapids. Maybe they're a little smarter down. I don't know.
Yeah, they are.
Then why did you come back here if they're smarter down there?
That's cool.
Oh, okay.
The basketball team's better than that.
All right. So
Yeah, no, they're not.
now you just keep doing this. Just keep crossing them. Keep the T's together. Keep the Y's together. Okay? So what type of T's are in this box? 2 capitals. What type of Y's are in this box?
1 lowercase, 1 capital.
So an upper and a lower. Okay? So this is where having a line over the top helps decipher whether it's a dominant or recessive trait. Yes?
Do you always have the capital letters first?
You should always put except for these, when you put them in the box, then put your capital letters first. Yes. But when you're outside setting it up, You want to go in the order that you see in your cross. Okay, so what do we have inside this box?
Okay. Capital T,
And,
lowercase y, lowercase y.
okay. Are we having problems deciphering what's going inside the boxes?
Nope.
No. Okay. So then what do we see here? What for our T's?
One capital, one lowercase.
Is that for both of them?
Yep.
Okay. Then what about the last one on the top? Now, one of the things you're not going to want to rush through here because it's very easy to make a mistake. Okay? Do you want to do the next 4 together, or do you want to work on these on your own?
I don't know.
Okay, so we'll go and hit pause again. All right, so moving on. Okay, what goes inside this box? Capital T and capital T, capital Y, little y. Okay? Then, do we possibly see a pattern emerging from this? Okay? Then, inside this box,
Cap T, lowercase c,
okay? And then capital T, lowercase t, okay? So then now let's get down into here, and then you're really perhaps going to see a pattern emerge. What do we see inside this box?
capital T, lowercase t, capital Y, lowercase y.
Okay? Then looks like capital T, lowercase t, 2 little y's. Okay? Then 2 lowercase t's for the first time. And then big Y, little y. Okay? Then looks like 2 lowercase t's. And then 2 lowercase y's. Okay?
That's the same as.
Then let's do our final one here, capital T, lowercase t, capital Y, lowercase y. Okay? Then capital lower, lower, lower. So capital lower, lower, lower. Okay? Then 2 lowercase t's, lower, lower, and then upper and lower. Okay? And then 2 lowercase, 2 lowercase. Okay. So the last thing we need to do for this problem then is, what is that asking? Yes, you could say it says phenotype, but what does that mean to you? Yes, what you can observe. So color is certainly something that you can see. This is the genetic makeup, all the different types of the genotypes. What you can see from this is your phenotype. I highly recommend that when you go through these, pick a box, and usually it's this one. What are the results that you would observe from this cross? This type of, let's say, the randomness of this, that's what genetics is you're studying the probability. That's what I meant to say. So this is all probability. This observable trait would look like what? Tall and yellow. So we just pick tall and yellow. And how many of those are we going to come across? Well, there's one. Just mark it in your box so you don't have to Otherwise, you might skip one, or you might count one of them twice. Okay? Do we see tall in yellow anywhere else? Okay? This one will be tall in yellow. Okay? So let's just put tally marks there. So there's 1 for this one. You said right below it, tall in yellow. There's 2. Okay? Below this one, okay, there's 3. And there's 4. Okay? Then this one's going to be what? Tall and green. Okay? So you said tall and green. There's 1. That's tall and green. There's 2. That's tall and green. There's 3. That's tall and green. There's 4. Like I said, sometimes you can pick up on different types of patterns that you can see. This one is tall and what?
Tall and yellow.
Tall and yellow. Do we have one of those?
We do.
Okay, so we'll put that here. There's 1. There's tall and yellow. Okay, what about this one? Short and yellow. So there's that one. And what about this one? Short and yellow. So we got 6 to 6 so far.
Why did you put short yellow on tall and green?
Short and yellow. Okay, so that must mean I got to take that one off and that one off. Okay, so short and yellow, there's 2. What about this one? Tall and green. Okay, so here we go. This one is tall and green. Mark him off. Okay, then we only have 2 left, and these are going to be what? Short and green and short and green. And there was what, 2 of those? Put that on the wrong one. Okay, so how many tally marks are here? We got 6. How many are here? 6. We got 2 and 2. Does that add up to 16?
No, because you forgot to. Yes, it does. It does. 12, 14, 16.
6 plus 6 plus 2 plus 2. 16. Okay? We maybe got a little bit of a late start, which is fine. We want to make sure that you understand this. Okay? So front row. Front row right side. Are we all right? Front row. Okay? Middle row. Back row. Okay? Back row. Okay. So I just want to give you a little bit of a visual aid here. One of the things now this is something to think about. Can sickle cell anemia be a devastating trait?
Yes.
Yes, it can. But if we can treat it, do you want to eliminate sickle cell anemia? No.
No.
Because, now this was something that they said in the video. That's why paying attention to it. Do individuals who have sickle cell anemia, will they die or have a chance of dying from malaria?
No.
No, they do not. So living in arid regions where malaria can be rampant, you don't want to eradicate sickle cell anemia because there's a type of resistance to it. That's why studying genetics is so important. I just want to show you a visual aid on this. This is where we have to stop this for a moment and we'll come back. So you can see why those type of mice get that name. It looks like they're shivering. They sometimes call a waltzing trait, or that was shiver a mice. So again, they were born that way. There's two ways that we can possibly treat that. One, with genetic engineering. Two, you can do stem cell research, which means a stem cell is kind of like a wild card when you're playing poker or whatever. A wild card just means you can make that into anything you want. Maybe another 9, maybe another king or something like that. That's what we mean by having a wild card. Okay, we'll pick up with this next week with your assignment. Make sure you get that vocab started. We'll probably start writing them up on the board on Monday, okay? Because they are due on Wednesday. All right. That's all I have. We'll catch up to you next time.